looking at fermentation in more detail. And the idea that oxygen is not involved, it's anoxic. And so if you are a facultative anaerobe or an anaerobe, you could be doing fermentation as an option. Hey, did you set up your anaerobe stuff yesterday in lab? Okay. So those are always interesting to see with the FTM tubes, as well as what you pop into the anaerobic jar and see what ends up growing on when you look at them on Thursday. And so this idea that, they're assessing their environment constantly, and they're trying to figure out what they have available and then deciding which pathway they should turn on to get the most bang for their buck. And so in that sense, they want to generate as much ATP as they can. And so even though they're faced with maybe a bunch of different chemicals to work with, they'll have some favorites. Fermentation, not so good. I mean, if I was a facultative anaerobe, probably wouldn't pick fermentation if I didn't have to. But sometimes you're forced into it and you have to run this pathway. So really though, the goal is to get some ATP, and in fermentation, it's again, you're looking at that substrate level phosphorylation, and they don't care about the product. That's not what they're going for. They don't care what that final product is. They just excrete that out. And in the industrial microbiology arena, we take advantage of that, with production of alcohol, as well as many other products. It's fermentation products that we are triggering in those microbes. They're excreting it, which is nice because they get it out of their cell. It's really easy then to take it and purify it. But again, it's not what they're going for. Even though we like, say, alcohol or whatever product they're making, that's not their goal. And in fact, it's like waste to them. They're poison. So that's why you can't ferment. Like if you make homemade wine, you can't push the alcohol content really high because that kills the yeast. And so it's waste to them. They're swimming around in their own waste. And so there's only so far you can take it. If you want to get more concentrated, then you have to distill and do it that way to increase the alcohol content. Right. So this is a generic example of substrate level phosphorylation, where it's at the level of the substrate in the pathway that you see the phosphate getting transferred from the substrate to the ATP in order to make ATP. And we've already looked at that with glycolysis, where you start with glucose, work your way down to pyruvate, and then what is E. coli, for example, doing once it gets to pyruvate? What does it start looking for if it wants the best ATP production? Oxygen. Yeah, it stops there and goes, okay, do I have oxygen? If not, it's like, bummer, I don't have any oxygen. Okay, I'm going to run some kind of fermentation ending to this pathway, finish it off with a fermentation section, and maybe it'll run the ethanol pathway. It depends on the microbe, though, how they're going to work this. And so here, this table is looking at different bacteria. Our column, you'll see the different species. And, I'm not asking you to memorize this. I'm just trying to show you diversity. There's diverse fermentation pathways. And you can see the ATP can vary a little bit, but it's really low. I mean, you go from 0 to 3 ATP. That's not great, but at least you stay alive. This is another way to look at it. So this, again, is not complete. There's all kinds of fermentation pathways there, but I like this one because it's really showing you that they can work with all kinds of different starting molecules. Like we tend to focus on glycolysis, talk about that pathway, so glucose, but you can see, so glucose is a hexyl sugar, that's what we've been focusing on, but you can see that they can take all kinds of different molecules and work it in a fermentation pathway. But again, it's just this kind of, throwing the pantry door open and going, what do I have to work with? That's what they're doing. They're assessing their environment. What do I have to work with? And then they'll put it into these different pathways. with the goal of making ATP. And so off to the right is what we've been talking about, this idea of generating ATP through these processes. And then, of course, the flip side is at some point, these microbes are also using ATP in order maybe to repair any damage that they're going on, increase their cell biomass. If they want to grow, they're going to expend some ATP. Okay, so we have the catabolism and anabolism ideas here. Right now we're focusing pretty much on catabolism, where they're breaking down substances to make ATP. So fermentation, that third phase that we were looking at with glycolysis, all those fermentation possibilities, you are seeing that there's no terminal electron acceptor in this process like we do with the electron transport chain. And so really don't get a ton of ATP through these methods, but it does at least get you something that you can survive off of. If microbes want to get a fair dose of ATP, they just have to keep running these fermentations a lot. And so that means you have to feed them a lot. So in the alcohol industry, you know, if you've taken beer tours or whatever, in those big, huge copper kettle vats of yeast, I mean, it's just a ton of carbon you're throwing out those yeast in order to get them to pump out enough alcohol to purify itself. And so large quantities of the substrate are needed in order to get the ATP they need to stay alive. They have to really crank it out. So no matter what pathway you're looking at, when you look at the fermentation pathways, there's a few key things. You know, we're talking about substrate level phosphorylation, certainly. But when you really look at all these pathways and go, well, why are they running that pathway? I mean, why are there so many different fermentation pathways out there? It's basically this idea of where they've evolved to live, what's around them. And then basically they're playing a game of running a pathway to have a balance of oxidation and reduction processes. So what we mean by that is if you go all the way from the substrate all the way to the products and look at what's going on with the electron numbers, generally the way to do this is we say that the substrate there, the number of electrons of the substrates has to equal the number of electrons in the products. You have to have this balance between the substrate and product electron number. So as they're running these pathways, the electrons removed from the substrate, they have to be given over to something. And so you have to end up in a situation where you have equal number of electrons, substrate to product. And so they often use the word disposed. These electrons have to be disposed of. Like, okay, I got to do something with this. What can I do with it in order to play the rules of the game, make sure I'm adhering to the rules of the game? And basically, it just depends where they evolved, how they're going to run this game. And so they will cobble together these pathways based on what they have available to them. But basically, when you look at all the pathways, that rule of the substrate electron number has to equal the product electron number. That rule is satisfied. And so if they can manage to do that, then they have a successful pathway. So this is given the fancy phrase redox balance. Redox balance. And all of these fermentation pathways have that. When you look at these fermentation pathways, many of them will utilize hydrogen gas as part of the process. And so basically it's the reason why is protons are usually fairly abundant around the microbe and in the microbe. And so it's really easy to donate off an electron to the proton and like here. And then that will make hydrogen gas. You know, you have two protons and two electrons reacting. And so it's just a really easy way for them to dispose of those electrons and hydrogen gas gets made. Another common strategy you offer is acetate or acetic acid will get me as well. And again, it's not that they're going for the hydrogen gas. That's not their goal. They're not trying to make acetate. They're just playing the game of, I need to dispose of these electrons so everything evens out when all is said and done, substrate to product. But basically, too, of course, all of this is allowing for ATP synthesis through that substrate level phosphorylation. Okay, so again, going back to all of these different pathways, okay, it's the idea that the substrate electron number has to equal the product electron. And if you play that game well enough, you can create a situation where you make, a few ATP molecules in the process. But the products, acetate, hydrogen gas, all of that, they don't care about. It's just, it helped them play the game successfully. Okay, so here are some aspects of fermentation where you can see hydrogen gas getting made, acetate getting made. So again, it's something where they don't care about the products, we do. Like here's a flow chart of some of the things we use the fermentation products for. Of course, alcohol is a big one. Most of us can think of that as a fermentation product, but butanediol, we use that in research in the lab, yogurt and cheese, butter, butanol, isopropanol, acetone. Okay, so all of these are fermentation reactions, super useful for humans. We make good use of a lot of this stuff. And but for the microbe, it's waste. It's actually something that they don't really want in high levels around them because it's waste. Okay, but certainly fermentation seems like fermentation is really gaining a better foothold now than it has in many, many decades. It's being popularized, which is good. Fermentation products are really good. As we get into human microbe interactions, I'll bring back the idea of fermentation products as food and how valuable it can be for our gut microbiome and for our overall health. But be aware there's a picture here. So all of these are fermented products. So that's why you can leave a tub of sour cream in your fridge and it takes a long time for it to spoil. It's because it's gone through the fermentation process. Where those dill pickles or pickles in general, most pickles on typical store shelves are not fermented anymore through traditional fermentation. Unfortunately, they don't use live microbes. They just pickle it in certain chemicals. So unfortunately. Okay, we'll come back to that idea a little bit later. But speaking of the gut microbiome, wanted to mention a few things that they talk about in this chapter. Interesting study on koala bears, where they were looking at different koala bears feed on certain types of eucalyptus. And if you give them the wrong type, they can die. And so they noted, however, that if they take a koala and give it a fecal transplant from the other koala that, feeds on a different eucalyptus, give it a fecal transplant, then that second koala can eat that other eucalyptus. And so again, pointing to the idea that the microbiome, the microbes of species, you've heard of fecal transplant, right? Where there's different ways to administer it, but basically getting feces, and it's not really that simple. They actually clean, while some places they'll purify the microbes out of the feces and create like a pill that you can swallow, or they might do with animals. Besides humans, they probably do this. But anyways, getting the microbes from the feces is essentially what we're thinking about here, and that now that second koala got the feces from the first koala, it can eat the first koala's food without dying. And so they did a study also with humans in Japan eating seaweed, nori. That's a big part of their diet, the nori. It's a type of seaweed. And it contains porphyrins as part of the seaweed. And if you eat raw seaweed, you're ingesting some of the bacteria that are on the seaweed, and they get into your gut, and they can break down those porphyrins. The bacteria that live on the seaweed, they use the porphyrins in the seaweed as a source of nutrition. So then they saw people that live in Japan and eat the seaweed, they're swallowing the seaweed plus the bacteria. And then they start to see that people in Japan that are doing that, they have bacteria in their microbiome, gut, that have this phenotype of being able to use porphyrins. And so basically what they saw to come to this conclusion that, okay, they don't see the bacteria, so they actually swallow the bacteria and out those bacteria that survive. But the gut microbes still had the ability to break down porphyrins. So as that initial bacteria came with the seaweed into the human, what they suspect happened was horizontal gene transfer, that you had that wild bacteria transferring in genes to use porphyrin to the normal gut. And when they looked for it, they found it. And so again, this idea of horizontal gene transfer, in this case, helping people that live in Japan to effectively break down the seaweed and use it efficiently. So this is looking at this idea of left. If you have the PUL, that is the enzyme that breaks down porphyrin. So if you don't porphyrins in the diet, I guess, if you want to think of it that way, versus where did it go? Oh, here. So I'm sorry, the yellow stretch there. So here they said the mice porphyrins, and what they were showing is that if you fed the mice porphyrins, then the bacteria that have the ability to break down porphyrins, their population level. So basically, it allows for the porphyrins to the rest of the bacteria to break down the porphyrins to allow them to really get to high levels, high populations in the mice. If you don't supply the porphyrins, those bacteria didn't really do much. Their population. So this is one of the earlier studies where they started playing with this. What happens if you feed the mice this versus that? And what happens to the microbiome under those conditions, all the bacteria in the mouse gut. Basically, and many studies after are showing again and again and again that when you eat what you eat, you are manipulating your gut microbiome. And so every time you eat, you are influencing who persists, whose population goes up, whose population crashes, and the ratio of different species in your gut. And so you have really a profound control on your gut microbiome, far more than we thought, just by the choices you eat.
So back at the,
Mm-hmm.
with the, when I have the low portion, would that be
Yes, right? Because if they're like, I don't have any porphyrins in my vicinity, I don't really want to maintain the genes for the enzyme to break it down. So they could purge. That could be a wise decision for the microbe to do that. Other questions? So I've had people ask me like, oh, well, what diet do you follow? And I tell them basically two things: an evidence-based diet, which means you have to go into the literature frequently because the evidence keeps getting updated and new findings, new studies. evidence-based diet and a microbe-centric diet. I think about what would make my microbes happy and what would keep them healthy and make food choices off of that. So we'll get into that a little bit more when we get into the human microbe section of the class. Here's another one, breast milk, human breast milk, and human milk oligosaccharides. Human milk oligosaccharides are a big portion of breast milk, but babies can't themselves directly break it down, which sounds weird, right? Basically, what the baby relies on is the right microbes in their gut to help break that down. And if they don't have the right types of microbes, then things can go bad. And so what some studies are showing is that Bifidobacterium, that genus of bacteria, is critically important for babies to have in their gut to help break down. And I like the catchphrase, Bifidobacterium benefits babies, BBB. And so they're finding that this genus in particular, if babies have enough of this in their gut, then they can take the human milk oligosaccharides and ferment it to lactate and do it in such a way that won't produce any kind of gas. And if you have ever had a baby and had to babysit, or one of my boys was colicky, it's not fun to have a colicky baby on your hands, literally. So again, this idea of fermentation pathways, which pathway are they running, in this case, can have big influence on the comfort of your baby, right? And so we try to minimize gas production in babies to minimize the colic issue. The Bifidobacterium in that study was also found to produce anti-inflammatory compounds that help the baby and beneficial immunomodulators. Human microbe. And then also with fermentation pathways, they're finding that neurotransmitters that we have in our body are the microbes are helping us with that. And so what they're finding is that when microbes are fermenting peptides and amino acids from the food that we eat, it can actually create products that they excrete, because remember, they're not going for the product of fermentation. They're just going for the ATP that gets made during fermentation. But our bodies can then take that and use them as neurotransmitters, some of those waste products. And so the production of dopamine, serotonin, histamine, they're all finding that, and GABA, that microbes can kind of kick off these neurotransmitter pathways based on their fermentation, the products they make, and then taking those and making neurotransmitters off of them. And so the gut bacteria are known to produce neurotransmitters that our bodies respond to. The same neurotransmitters that we've been talking about in neuroscience, it's gut microbes making many of those. And so here they're showing GABA and histamine being produced. And so they're really kind of looking at this more carefully now. You might have heard the catchphrase gut brain. Our gut is actually like a second brain in a way. So I'll talk more about that in the human microbe section. But really, really interesting research coming out now on how integral and important the gut microbes are. Okay, so we'll get into that in more detail. One little sticky wicket I wanted to mention here. So fermentation, I keep telling you, it's substrate level phosphorylation. There's an exception that I wanted to mention. So this is fermentation without substrate level phosphorylation, but technically when you look at it, it's fermentation. So what we mean is that some reactions, there's a handful out there, where when you look at the pathway and you do the energetics of it, calculate the energetics, there's not enough free energy to make ATP from it. What's the magic number? Negative what for ATP? Negative what? 31, I think your book says -31. A lot of other sources say -32. It's like 31.8 or something like that. I round up. Apparently your book rounds down. But anyway, so they didn't, when you look at the pathway, it didn't get you -31 free energy kilojoules per mole. And so, but they still managed to make ATP. Can you imagine how confusing this must have been when they had to try to figure this one out? And so when they studied these bacteria more closely, what they discovered is that they're using some kind of ion pump. And it can be protons, but they've also found some microbes, depending on their environment, they might use sodium, which is also a cation. And they can show that the microbe is pumping these, either the proton or the sodium, across the membrane. And as that's happening, it sets up, if you do it often enough, it sets up a proton gradient or sodium gradient. And then they can link that back as the protons or sodium diffuse back across the membrane. It has to go through the ATP synthase, and ATP gets made. So this is really weird because it was, I think, one of the first reactions was this one, where they were studying bacteria in the lab. They fed them succinate, and they figured out that the microbes were doing this reaction. Succinate in water goes to propionate and bicarbonate. And if you do the free energy calculation, it's -20.5. That's not enough. We need -31. And so they must have been really scratching their heads going, but we still see ATP. The bacteria live, they reproduce. Obviously, they're getting their ATP needs met. So they had to hunt for, okay, what the heck are they doing in order to get at least -31? So they started looking more carefully at these bacteria, and they found the pumps and started to track that a bit better. And so they could link that there are sodium, in this case, bacteria that use sodium. And so it had this enzyme in the membrane that could act on succinate and, run that pathway where you go from succinate to propionate and bicarbonate. As that reaction is happening, it allows for sodium pumping. And then you have to imagine sodium. I wish the artist had been a little more extravagant, but put more sodium up here, sodium, sodium, sodium. And then you get a gradient of sodium across the membrane. So as the sodium diffuses back, it has to go through in this case, it's a sodium ATPase. And through that process, you'll get ATP. So technically, yes, this is a fermentation. Succinate in water. Technically, yeah, that's fermentation. But they link it to, in this case, sodium pumping to create a sodium gradient. So some microbes are wacky. They can do amazing things. So this is a good example of an exception to the rule of that substrate level phosphorylation. Okay, questions on any of that? Okay. Well, I'm going to get into the next slide set on more of the metabolism. Okay. We've seen these pictures already, substrate level phosphorylation versus oxidative phosphorylation. And so with the oxidative phosphorylation, they're just trying to show that you have an electron transport chain in the membrane and you develop a gradient, a proton gradient, which allows for ATP synthesis. So we're going to get into that a little bit more and look at how microbes might have to tweak that. So you've seen this already, where we have an electron transport chain process with, what's the terminal electron acceptor here? Can you find it? Yep. You see the oxygen and the terminal space there. Okay, so oxygen is the terminal electron acceptor. As the electrons are flowing from donor to acceptor, through that process, you get key moments where there's a little bit of extra free energy available to pump protons across the membrane. And so you get that linking of electron transport chain process to proton pumping. And then that proton gradient allows for ADP and inorganic phosphate to react as they flow through the ATP synthesis. Right, so we've been through that. So now let's think of a situation where a microbe is hanging out in an environment, maybe like in the anaerobic jar in lab. And so you have forced that microbe now to very quickly be in the acid phosphate. Those chemical packets that got put into the jar are scarfing up all the oxygen. And so now you're forcing them to be in an environment without oxygen. So now this is going to fall apart, so to speak. They can't do this. And so at least one of the microbes in your jar should be able to pivot, so to speak, and go, okay, my oxygen's gone, but I can do something else. I can still do some version of this and put something else at the end of the electron transport chain besides oxygen. So we're talking about anaerobic metabolism. Typically when we say that phrase, anaerobic metabolism, we're typically thinking about an electron transport chain process. It's just now we don't have oxygen as the terminal electron acceptor. It's some other molecule that's available in the microbe's environment that's handy to stick in there as the terminal electron acceptor. And depending on the microbe and where they're living, it can be some kind of inorganic molecule that they'll stick at the end of the chain, or maybe it's organic. So you'll see some version of an electron transport chain. There might be slightly different players in the game, but it's still the overall process of the ETC. However, when you run the energetics and look at what the payoff is, it's not as much energy compared to if they did aerobic respiration. So remember, oxygen is the best electron acceptor. Anything besides oxygen, you don't get as much ATP. So again, if you're a smart microbe, you'll pick oxygen. But in that anaerobic jar, you're forcing them into, now I got to pick something else. And hopefully there's something else available that they can use. So again, they don't care about the product that gets made at the very end of the electron transport chain. That's not really what they care about. They'll just excrete that. They don't care about it. They're just creating a situation to make a proton gradient. So going back to the redox tower, we're looking at, okay, well, it's not oxygen. At the bottom. Remember, the further the electrons fall on this tower, the more ATP you can produce. So you can see oxygen's always at the base, but then there's other ones that are down there. You see NO-, you see Fe+. So those would be good options. If I was a microbe, that's what I would be hunting for. I'd look for something like that. And if I have the right donors and acceptors set up in my membrane, I can just go ahead and run that pathway instead, that electron transport chain with maybe nitrate, NO3-. Stick that at the end. And so a lot of microbes do that. And we'll look at specific microbes, I think it's next week, maybe, or is it? I think it's next week, where we'll see if they can do this particular reaction. And so we're looking at the idea of nitrate, NO3-. So if they stick that at the end instead of oxygen, what they will do is run a reaction where they might, they'll go from nitrate to nitrite. And then we're gonna find in labs, that's as far as some microbes can go. Like that's the only thing they can do. They can't take it any further. But there's other microbes that can take nitrate and keep going and going and going and going until they end up with dinitrogen, N2. And again, their goal is not to get N2 or nitrites, not what they're going for. It's just creating a situation where they can do this electron transfer chain process and get their proton gradient, which leads to ATP. And so it's just the game they're playing. They just excrete all this. It's important to realize that the last product here, nitrous oxide, and dinitrogen are all gases. Is it one of those laughing gas? Is it nitrous? Nitrous oxide? It's either nitric or nitrous. Does anyone know? Laughing gas? Do they still do that in dental offices? I'd much rather have that than getting an injection. So this is, if those gases are made, then they just get excreted out, and those get, that's part of the reason why we have dinitrogen in our atmosphere. It's microbial origin that's allowing for that. And so microbes are really good at recycling. In this case, we're looking at nitrogen containing compounds, getting it from nitrate into the gas state. And so they're really good at recycling like that. So nitrate is inorganic. So inorganic nitrogen compounds are pretty common as electron acceptors because there's usually some inorganic nitrogen around for microbes to take advantage of. And so you will see plenty of microbes that can run these reactions. So for example, E. coli can take nitrate in an electron transfer chain. I'll put nitrate at the end. And the product is nitrate. And they just excrete that. They're like, yeah, I don't care about that. It's just making a situation where I can get my ATP. Pseudomonas, on the other hand, can take it all the way to dinitrogen. And so it just depends on the microbe and what complexes they have in their membrane. So here on the left is aerobic respiration, where you have oxygen as the terminal electron acceptor. But if in that anaerobic jar, you created no oxygen, and so maybe the microbes are going, well, all right, no oxygen, but I know there's nitrate. I can take that, stick it at the end of my other electron transport chain. And you can see it's still electron transport chain process, slightly different players in the game. But overall, it's still the situation where you have electron transport chain linked to proton pumping. How many protons are pumped on the left side in that image? How many? 5. How many on the right? 4. So the artist is nodding to the idea that, yeah, you don't get as much proton pumping if you have nitrate in place of oxygen. So basically, if you look at any of these pathways, when you have oxygen at the end, you get more proton pumping. If you have anything else besides oxygen, less proton pumping. So that's why you get less ATP being made under those conditions. So here is another one where their Pseudomonas can take nitrate and just keep going and going and going all the way to dinitrogen. So that's interesting here. But again, it's just an electron transfer chain process where you have all these complexes embedded in the membrane, donor to acceptor on down the line until you can wrap it all up, in this case, all the way to dinitrogen. And so if they can piece it together like this, typically it allows for a little bit better proton buildup, proton gradient buildup, if you can keep it running. And so Pseudomonas stutzeri has been well studied for this. Now, I wouldn't want Pseudomonas in my soil if I'm growing plants. I wouldn't want this going on because plants love nitrate, which you want in the soil. But if you have a ton of Pseudomonas in your soil, they're doing this in your soil. Then your plant is denied nitrogen. So again, even if you are, a farmer or grower working with plants, if you understand the soil microbes, you can maximize your plant growth by understanding what kinds of microbes you want in your soil versus the ones you don't want in your soil. You don't want them taking away the nitrate that the plants love. Okay. So again, another plug for if you have a little bit of microbiology training, it's apply the microbiology training in so many different directions. All right, I guess we got to stop there. We'll stop there and continue this on Friday.